Okay, hi everyone. Thank you for coming to our December Lunch and Learn. My name is Maysen Mesaros. I'm the new genetic counselor for the neuroscience department at the Medical University of South Carolina. And today I'm going to be talking to you about genetics and genetic testing in Parkinson's disease. So I just wanted to first give a little bit of background about myself. As I said, I'm the new genetic counselor here. I'm the first one in the neuroscience department. So I just moved here to Charleston from Ohio. I went to the Ohio State University, which I hesitate to say because sometimes I get booed here. But I studied molecular genetics as my undergraduate degree, and then I just graduated with my master's in genetic counseling from Ohio State as well. I support all of the neuroscience department at MUSC, so that includes movement disorders and Parkinson's disease, but I also see neuromuscular disorders, stroke, epilepsy, sort of everything under the sun. So before I talk about genetic counseling in Parkinson's disease, I think it's only right that I first give a little bit of background on genetic counseling in general. So the formal definition that's put out by our national society is that genetic counseling is the process of helping people understand and adapt to the medical, psychological, and familial implications of genetic contributions to disease. So that's kind of a wordy definition in my opinion. But essentially my job is to help find a genetic explanation for neurological disease and help patients understand that. So help patients understand their genetics, what that means for their health and for their condition, what it could mean for their family members, and then also helping them adapt to that information. And sometimes there can be psychological impact from some of this genetic information. And so my training includes both science and genetics, but also psychosocial aspects. Something that I think is important to highlight is that genetic counseling does not always equal or lead to genetic testing. So my job is to meet with patients and talk through the option of genetic testing, see if it's right for you, if it would benefit you, if you want that information, but not every single patient that comes through my door goes through testing. So sometimes it's just a conversation. As you probably have assumed, genetics is a whole family affair a lot of time because we're talking about heredity. And so my role as a genetic counselor involves supporting both the patient in front of me, but also family members. And I'll talk more about that later on. So what a genetic counseling appointment would look like with me or another genetic counselor in adult neurology is you would be referred to me by your neurologist. And when you meet with me, I ask questions about your personal history, your family history, try to get all of the patterns and information that I can get to lead me in the direction of, is this hereditary or not? Then we'll talk about the genetics of Parkinson's disease. And then ultimately, sometimes that leads to genetic testing, which can be a patient getting their blood drawn when they come to see me. We have saliva kits, we have cheek swabs. Those things can even be sent to patient homes if you're not able to come see me in person. So there's lots of ways that genetic testing can be done and ways that we can collect samples. So before we talk about the genetics of Parkinson's disease in detail, I want to give a quick little crash course on genetics because for some people, it's probably been a while since you've learned this information. But our genes are like the instruction manuals for our body. So our genes are made up of long strands of letters, which is our DNA. And those letters spell out instructions for how our body should be functioning and behaving and developing. We all have the exact same genes. So everyone in this room, everyone on this call, we have the same genes. It's just a matter of, are they spelled correctly or not? Are they working, doing their job or not? We also all have 2 copies of every single gene in our body because we get a copy from our mother and a copy from our father. And so when someone is born with a mistake in the instructions of their gene, we call that a mutation. And those mistakes can lead to disease because your body gets confused. The instructions no longer make sense. And that can lead to diseases such as Parkinson's disease. And that's what genetic testing is looking at, is whether or not your instructions are spelled correctly. And when we identify a mutation, we can talk about the implications of that, what that means for your health and for family members. So the reason I mentioned that we have 2 copies of every single gene in our body, one from our mom and one from our dad, is because some genetic conditions require that just one of your copies is broken. So you just have one mutation. And other diseases require that both copies, the copy you got from your mother and your father, are broken. And so that'll be important later when we talk about some of the genetic forms of Parkinson's disease. And this is just a visual to show you what our genes and DNA look like. So if you zoomed in very closely on a gene, when I said the long strands of letters that spell out instructions for our body, this is what that looks like. And so genetic testing is looking at your specific instructions or letters to see, is this the correct spelling, or where there's a T, is there an A instead? Is there a spelling mistake, which we call a mutation? So there's a couple different ways that genetic diseases can happen. And so the one we're going to focus on mostly today is called monogenic. And so that means that there's one mutation in one gene, and that's enough to cause disease. And so that's what genetic testing, when you come to see me in a Parkinson's disease clinic, is looking for, is a monogenic cause of Parkinson's disease. There's also polygenic disorders, which means that sometimes there's several mutations in several genes that combine together and cause an additive effect to cause disease. And sometimes those additive effects even combine with environmental effects, which is called multifactorial disease. And so Parkinson's disease in general is considered a multifactorial condition, meaning we know there's some genetic components and we know that there's some environmental components. Some of the time we can identify a monogenic, meaning single gene, single mutation cause, but most of the time it's a polygenic or multifactorial disease where we cannot actually identify that single genetic explanation. And so this is just another visual showing sort of the different causes of Parkinson's disease. We have identified genetic risk factors. So those are the monogenic causes that I talked about. And that's what we do genetic testing for. That's a single gene, a single mutation that we can say this was a strong contributing factor to your Parkinson's disease. But there's also the unidentified genetic and environmental factors that mix together and sort of create this cocktail to cause disease. And then the known environmental factors, things like insect repellents and paraquat that we know have an increased risk factor for Parkinson's disease. So some of these are helpful. So I know, not that I'm endorsing smoking, but there have been studies that say smoking cigarettes is a protective factor against Parkinson's disease. So some are helpful, some are harmful. I don't know the specifics of all of them. I think that caffeine is also helpful. Which one? Yeah. I don't know the specifics about alcohol, if that's harmful or hurtful. I'm not endorsing any of these things. Oh, okay. Okay. Yeah, so the question was, some of these environmental factors that you can see on the right, are they protective factors? Are they harmful factors? From my understanding, I think a few of those are protective. I've heard that smoking cigarettes, not that I'm saying you should do this, but there have been studies that say that can reduce the risk of Parkinson's disease. Same with caffeine. but I don't know the specifics of every single environmental factor here on the right. This is just to show that Parkinson's disease is a combination of environmental and genetic factors, and we're just now starting to sort of dip in the single genetic causes, the monogenic causes that we talked about. And that's what genetic testing with me would look like and what it would look for. So a monogenic cause, meaning we do genetic testing, we find a single gene, a single mutation that we can say this led to your Parkinson's disease. That is identified around 10 to 15% of the time. So the other 85% is still unclear. We know there might be some genetic components in there, but we don't have the technology yet to pick it up. We're only picking up around 15% at this time. And so I use this visual for multifactorial disease, or this analogy, if you will, about a jar of marbles. And so if you look on the right, you can think of someone starting with this many marbles in their jar, and maybe each of those marbles is some sort of genetic factor. And then as you add environmental factors to that, maybe you grew up on a farm, or firefighters that are exposed to toxins, and you add more marbles to that jar, eventually it's going to fill up. And when it's full, that causes disease. We do not have the technology right now to test for every single marble in that jar and say this plus that led to Parkinson's disease. We are only as smart enough right now to find a single marble that we know has a really strong risk factor, and say this was a strong genetic component to your disease. So why would someone consider genetic testing for Parkinson's disease? A main reason that I see for a lot of my patients is for clarity and for answers. And so some people just want to know why this happened. Sometimes we see that it runs in a family, and it can have a lot of impact on your life, of course, on family relationships, and to have closure or clarity as to why this disease is affecting the family can be helpful for people. Sometimes it can assist with prognostic information. So sometimes when we can identify a genetic explanation for Parkinson's disease, we can say, well, this specific genetic mutation is associated with cognitive defects. And so maybe we need to have you evaluated for cognitive decline, or this specific mutation is more so responsive to levodopa. So let's try levodopa, or it's not responsive to that. So let's not do levodopa. Sometimes it can give us information about prognosis. So is this a rapidly progressive type of Parkinson's disease, or something that might be more slowly progressive? So those are all things that we're starting to get out of genetic testing, as more information for people about the specifics of their disease. Additionally, this is newer, but it can be helpful for clinical trial eligibility. So in medicine in general, we're starting to try to treat the underlying genetic problem of diseases instead of just treating the symptoms as we see them. And so, for example, instead of treating Parkinson's disease with levodopa, maybe we could actually target the underlying genetic problem. And if we can target that and fix that, that might have a greater benefit on treating the disease. And I'll give an example of one of the clinical trials that exists at the end of this presentation. Family risk assessment is another big one. So lots of people want this information for things like children, wanting to know, are their children going to develop Parkinson's disease, their siblings? And genetic testing is one way that we can get more information about that. It's not a perfect risk assessment, which I'll talk about, but it can give us more guidance to talk to the family. So as I mentioned, that single gene monogenic cause is only identified in around 10 to 15% of cases right now. There's a few different genetic testing panels, we call them, which means we're looking at several genes that we know can cause Parkinson's disease. And so these panels are offered in several settings. If you see an adult neurology genetic counselor like me, There's also a research study called PD generation, where genetic testing can be done to essentially do the same thing that I do, but it'll also connect you with research registries. And the overall goal of something like PD generation is to eventually get to the point where we have targeted therapies and treatments for these underlying genetic causes of Parkinson's disease. Anyone that, oh, go ahead. Mm-hmm. That's a great question. So the answer is no. Oh, I'm sorry. So her question was, if you had genetic testing done through 23andMe, does it look for all of these genetic causes of Parkinson's disease? And so the answer is no. So 23andMe, I believe, has LRRK2 on there, which is the LRRK2, which is a common cause of classic onset Parkinson's disease. The problem with 23andMe is 23andMe looks for very specific genetic changes that are found in the population when there are thousands of genetic changes out there. And so they might look at 5 of them and say, okay, you're negative. We didn't see a mutation, when really they need to read through your entire gene to see if they can find a mutation. Even if you did have positive genetic testing with 23andMe, you still need to get that clinically confirmed. So that would mean seeing someone like me, where we would essentially repeat testing in a clinical setting to be able to use that information for your clinical management. Yes. Yeah. That's a great question. So, it depends on the specific gene, and I don't have them all memorized, but I believe LRRK2, the problem is it's overactive and it's creating too much protein. And so one of the targeted therapies that's in clinical trials right now is trying to target the LRRK2 protein in the brain and get rid of it because there's too much of it. And so LRRK2, I believe, is an overactive problem, but there are some genetic changes where the gene is turned off or it's not creating protein. It just depends. And I forgot to repeat that question, but the question was, for these genetic changes like LRRK2, is this causing a problem where it's creating an overactive protein or creating too much protein or not creating the protein at all? This is all information, though, that we get when we order genetic testing. So when I get a result back and I see that someone has a mutation in LRRK2, then I can say, okay, this is an overactive mutation, or maybe this is something where someone's not making enough protein. That's all information we get on the back end. Yes. Yeah. Yes. Yes, it is. I will. I think. Is it, hello? Okay. So the question was the alpha, I can't say the word either, synuclein, protein. Is that one of them that's tested with traditional genetic testing for Parkinson's disease? And the answer is yes. So that SNCA gene that you see there causes that protein. Another thing that I wanted to address is cost of genetic testing. So right now there are a lot of sponsored programs where anyone that has a diagnosis of Parkinson's disease is eligible for genetic testing, and it's at no cost. So either if you did that through PD generation, or if you were to come see me and not do that through a research study, either way, you can get no cost testing, which is a really wonderful thing about Parkinson's disease genetic testing right now. So it doesn't even go through your insurance at all, because it's sponsored by pharmaceutical companies that are trying to come up with drugs and therapies to target these genetic problems. Some labs that we use, we can go through Medicare and not do sponsored testing, and usually costing is no more than $75 if we go that route. So most monogenic causes of Parkinson's disease that we talked about, so single gene, single mutation, are inherited in what's called an autosomal dominant manner. And so if you remember, we talked about some genetic diseases require that just one of your copies, either the one you got from your mother or your father, is broken. Other diseases require that both of them are broken. The majority of classic onset, adult onset Parkinson's disease is inherited in a dominant manner, which means that just one copy is broken. So if you see this visual here, that's showing 2 copies of a gene that causes Parkinson's disease. One of them has a mutation, and that's enough to contribute a strong risk to Parkinson's disease. When someone has this, there's a 50-50 chance whether that broken copy is passed on to a child or not, because you either pass on your good one or your bad one. And so if this were identified in a patient that I saw that has Parkinson's disease, we would then know that there's a 50% chance for children to also have this risk for Parkinson's disease. Now it gets a little bit more complicated than the 50-50, because not every single person that inherits one of these risk factors will develop Parkinson's disease. So we know that they're strong risk factors, but it's not a 100% risk. So for example, for LRRK2, most of the time, there's only a 32% chance that someone's going to develop Parkinson's disease by the age of 80, even if they inherit one of these mutations. And so that's why I said when we talked about earlier family risk assessment, it's not perfect and clear cut. I can't say your child's going to get this or they're not most of the time. But we can at least get more specific with numbers and maybe say you have a 32% chance. And that's all information that it's up to the patient if they want that. It's up to my patient's child if they want to come in and talk to me and learn that information. But just knowing that it's not black and white. So these are the 2 most common genetic causes of the adult classic onset Parkinson's disease. So we talked about LRRK2 a little bit, and you may have heard of a gene called GBA. And so these 2 are always genes that should be tested for someone that has a classic presentation of Parkinson's disease. But you can see in the comment section for GBA, for example, it's associated with increased risk for Lewy body dementia, or you might have more atypical motor findings associated with a GBA mutation. And so this is an example of when we might find something from genetic testing and be able to tell you, potentially what to expect going forward, or do we need to evaluate you for other types of symptoms to best manage your disease. Something unique about the GBA gene. So this is an example of a gene where when you have one broken copy, that's enough to cause disease. So that's an autosomal dominant disease. However, when people have 2 broken copies, so both of their copies are broken in GBA, that causes a completely separate disease called Gaucher disease. So Gaucher disease causes problems with your bones. It causes bone pain. It can cause anemia. It can cause your organs to be enlarged, like your spleen or liver. And so sometimes people will have a family history of this disease, and then others will have Parkinson's disease. And so again, the importance of me asking you family history information, knowing if anyone has Gaucher disease can be really helpful to know, is there a GBA mutation that's running through the family? Sometimes, so Gaucher disease is an example of a disease that people, before they decide to have children, sometimes consider carrier screening. And that means figuring out what diseases they have the potential to pass on to their children. And Gaucher disease is one of those. And so sometimes people find out they have this increased risk for Parkinson's disease, and they weren't looking for it. They were trying to figure out for reproductive purposes what their children could potentially have. So sometimes it can be an incidental finding in the reproductive setting. And then this is just another visual just to show you all of or most of the known genetic causes of Parkinson's disease right now. And so some of them are associated with earlier onset Parkinson's disease. So the SNCA gene that we talked about. There are some that are associated with more juvenile onset. So I wouldn't expect to see that when I have a patient with classic adult onset Parkinson's disease. So I want to talk about the limitations to testing, and some of them I've already alluded to. But I think the most important thing is that we are very much still learning about the genetics of Parkinson's disease. And so even in the last few years, we have come a long ways. But we talked about only around 15% of the time right now are we identifying a single genetic cause that has a strong risk factor for Parkinson's disease. And so that other 85%, sometimes I see people that have a family history. They have a parent and a grandparent that had Parkinson's disease, and I test them and we don't find anything. There's still probably a hereditary component in there or a genetic factor. And maybe it's like that marble analogy where there's 10 marbles mixing together that are causing disease in this family. But we just can't identify that right now. And so it can be difficult to then give risk assessments to these individuals. I can't say your children are off the hook. They still might have a slightly increased risk for Parkinson's disease. My hope is that eventually we'll have something like a polygenic risk score, which we're starting to develop these for diseases like cancer, where you can look at several genetic factors in someone's makeup and say, this is your risk for cancer. So my hope is that we'll get there with Parkinson's disease and say, because of all of your genetic factors, this is your lifetime risk to develop the disease. Also, genetic testing does not take away or change a diagnosis. So a lot of times patients will come and see me and we get a negative test and that they think, okay, well, then I don't have Parkinson's disease. And that's not the case. So this is just looking for the underlying why. But it doesn't take away a diagnosis by any means. And we already talked about the risk assessment not being completely concrete for family members, especially if someone tests negative. But even if they do test positive, we might be able to say your child has a 50% chance to inherit this risk factor. And even if they do, they might have a 30% chance to ever get the disease. So it can be very complicated. And that's why it's important to meet with a genetic counselor, because every patient and every scenario is different. And, talking through that with everybody is important. Yes. That's a great question. So his question was, is there any evidence that this is sex linked, that these genetic causes are sex linked? So there are some diseases that can be X-linked or sex-linked. The majority, I personally don't know of any causes of Parkinson's disease that are sex-linked. So these are autosomal diseases, meaning they're on your chromosomes that are not your X or your Y chromosome. So LRRK2, GBA, SNCA, those are all genes that we all have regardless of your sex. Yes. Ashkenazi Jewish, yes. That's a good question. So her question was, a lot of these genetic causes of Parkinson's disease seem to have higher rates in the Ashkenazi Jewish population. And so if you find from 23andMe that you have a relatively small portion of Ashkenazi Jewish ancestry, does that take these things off the table? And so with the Ashkenazi Jewish population, there are a lot of what are called founder mutations in that population. So known mutations or spelling mistakes that exist in the Ashkenazi Jewish population. And because it's such a niche group, we know that they have a higher risk to have mutations and things like LRRK2 and GBA and lots of other diseases that are not related to Parkinson's disease. Usually when someone has a pretty high portion of Ashkenazi Jewish ancestry, like more than 25% or more than 50% is when we start to think you might have a higher chance of having one of these mutations. But just because you don't have a lot of Ashkenazi Jewish ancestry does not take those mutations off the table, which takes me back to your question about 23andMe and does that look for all of these genetic causes? And 23andMe usually looks for those founder mutations. So that's really not going to be helpful if you're not of Ashkenazi Jewish descent, because if you're negative, you can still have a mutation in those genes. Does that answer your question? Okay. So the family risk testing, which I've talked about a little bit already, but a lot of times when I have a patient and they test positive, they then want to have their kids tested or have their kids come talk to me to see if they're interested in getting tested. We've already talked about how having one of these mutations does not guarantee you're going to develop Parkinson's disease if you are asymptomatic. If a mutation is not identified in my patient that has Parkinson's disease, it is not helpful to test their children. So sometimes I'll have a patient in front of me that's negative and they'll say, okay, well, should my kids get tested? But that's not going to be helpful because until we identify the genetic defect in the family that's causing Parkinson's disease, I'm not going to be able to interpret your child's negative result because it could just mean we haven't discovered the genetic defect in the family yet. So it's usually best to test the affected person and find the defect first if we can, to then be able to give informative information to family members about their risk. So when patients test negative and want to know information for things like their siblings or their children, I usually have to just go off of population risk alone. And so we know that the general population risk for Parkinson's disease is 1 to 3%. And we know that when someone has a parent or a sibling with Parkinson's disease, their risk is 2 to 3 times greater than that 1 to 3%. So when I see a negative genetic test, I usually quote around 12%. I list the highest number until we can learn more information about the genetics of Parkinson's disease. So this is that study that I was talking about that is targeting LRRK2. So this study is sponsored by Biogen. It's called the REASON study. And so essentially it's trying to reduce how much LRRK2 is produced, and it's trying to block the production of this protein. And so your question, sir, about, what are these genetic defects actually doing to the protein? This is making too much LRRK2. And so we're trying to reduce how much protein is being made. So a lot of neurological conditions, the problem is a toxic proteins being made or too much proteins being made. And so the direction we're heading is trying to create treatments that can target those toxic proteins or get rid of too much of the protein. And so we're getting there, but it's not easy because one, a lot of these problems are happening in the brain. And so it can be difficult to come up with a medication that can cross the blood-brain barrier and actually target where the problem is happening and do that in a safe way. We also don't want to get rid of all of your LRRK2, for example. So if there's too much, we want to get rid of some, but we can't get rid of all of this protein. It's a protein that we all need. So that's why these trials and these genetic therapies are taking a while, because we have to do it in a safe and very, very targeted way. And also everyone's body looks different. And so we're starting to get there. My hope is that we continue to get there and continue to progress. But we're still very much learning about the genetics of Parkinson's disease. I think even if we don't have a therapy now, a lot of patients just want that information to say, okay, I know I have a GBA mutation and maybe in 5 years there will be a therapy for that. And so some people are comfortable gaining that information now and knowing that maybe in the future we'll have something specific for it. Okay, and that's all that I had, but I'm happy to take any questions. Yeah, that's a good point. So he was saying that, lots of times genes are influenced by other genes. And so has someone must have looked at, is there another gene that is having an influence on LRRK2? And are we targeting that gene? And so that is another reason why this is so complicated, is because it's a network of so many pathways. And there are some genetic conditions where we know there's one gene involved and we can just target that. But something like Parkinson's disease, where we know it's this marble analogy where there's maybe multiple genetic factors and environmental factors, it's a lot harder to come up with a drug that's just going to knock out the extra LRRK2. We still might have a problem upstream of LRRK2. That's a good point. Yes. Yes. That's a great question. So her question was, is paraquat, that has been proven to have an increased risk for Parkinson's disease, altering DNA? My assumption would be probably. So I think even if you're not born with a genetic defect that causes Parkinson's disease, a lot of diseases happen by outside factors altering your DNA. So it's not something you have from the get-go, but some sort of toxin may have, altered this. And so it's possible that it could alter maybe one of the genes or one of the pathways, or one of the proteins, but there's not really strong information saying paraquat does this to LRRK2, for example. So one question is, how do I qualify for the Biogen study? So from my understanding, they are not currently recruiting. I think it is currently ongoing, but there's a website called clinicaltrials.gov, and the recruitment information is on there. So if they do open it back up to recruitment, that will be there. That's definitely something that I can look further into, but the last I checked, it's not an actively recruiting study. Yes. So, and the PD generation that I talked about, the genetic testing that you can do through there, and that sort of connects you with research opportunities, that's through the Parkinson's Disease Foundation. And so that's a good one to do. We are not associated with PD generation at MUSC, but I can help you connect with that, because the benefit of that is being connected into those research registries where if something does come up, you can be easily contacted. Not for PD generation. So they do it through a clinical laboratory. I believe they do it through Fulgent Genetics, which is a clinical lab. So good question.
I'll send out information in my thank you email.
Okay. Okay. It was probably something different, but they're all similar in a way. But the PD generation will be clinical based testing, and then they'll connect you with a genetic counselor, which is what I am, at another site that can talk through the results with you. So Nicole said she's going to send out information for the Parkinson's Disease Foundation and how to get connected with PD generation in the thank you email. Okay, so one question is, do I have a lot of patients that have Parkinson's disease that showed up as negative on 23andMe and test because of the incomplete testing on 23andMe? So I personally have not encountered that, but I am pretty new in my position, but I do know that a lot of my colleagues have. And some colleagues have mentioned that their patients have had positive testing after they had negative 23andMe. So, 23andMe can be very interesting and can be helpful if you identify something, but a negative is definitely not a true negative by any means. And no matter what, you should always have clinical testing. Good question. The other environmental factors like mold that can cause Parkinson's disease. So this is a little bit out of my wheelhouse. I'm not exactly sure. The only one that I know is proven is the paraquat. I do see it. I don't want to put this out there and say that it's true, but I do see quite a few firefighters with Parkinson's disease. I don't know for a fact if there is a correlation there, but I think we're behind on studies of environmental factors in Parkinson's disease. Okay, so the multi-system atrophy, there's a question that says, have we found genetic information related to MSA and Parkinson's disease? So MSA, usually no. It'd be really unlikely that we would find a genetic explanation for multi-system atrophy. There are a few studies that have suggested some unique mutations, but it hasn't been replicated. It's still something, especially if there's a family history, that I would be willing to meet a patient for and see if there's any testing that we could try, but it would sort of just be under the caveat that it would probably be a very low yield that we would find anything. So definitely with the MSAs, with both the Parkinson's related and ataxia related, we are behind on that. Yes. Oh, yes. So the question was, do you need a referral to be able to meet with me? So yes. So if you are set up with a neurologist, all they need to do is just refer you to genetics, to neurogenetics at MUSC. And that's it. So you can just ask them to put in that referral. It's pretty simple, especially if you're followed at MUSC. They all know me and I can get you right in. But that's the easiest way to do it. Yes. Yes. Yeah, so the question is, people that have a genetic predisposition that we identify through genetic testing, do they deteriorate or progress faster than those that have, let's say, an assumed environmental exposure? And it depends on the specific genetic defect. So there are some that are more quickly progressive. I believe VPS35 is an example of a gene that's quickly progressive, where LRRK2 is more slowly progressive. So it really just depends on the gene it would be identified in. Yes. So that's a little bit out of my wheelhouse. This question was those that have been diagnosed and potentially related to Agent Orange in Vietnam. Is that a progressive or slowly progressive course of the disease? That is out of my scope. I don't know the answer to that. Yeah, I don't know if there's any good data on that either. Yes. Uh-huh. Yeah, so his question was, is there any relationship between essential tremor and Parkinson's disease, especially when there's a family history of essential tremor and then others have Parkinson's disease? The answer is maybe. I'd say the most likely scenario is that maybe those were Parkinson's disease that were misdiagnosed, but usually essential tremor is not a hereditary disease. So if it truly was essential tremor, I would say probably not. But if it was a misdiagnosis or maybe an early sign of Parkinson's disease, then I would say there could be a connection. Any other questions? Is there any on here? Yeah, so someone stated that they have Parkinson's disease and a brother that passed away from multisystem atrophy. So I think that, that would be a good candidate for genetic testing, even though the MSA, we don't have a lot of good information right now. It still could be something where we could try to test and see if there is some sort of connection. There is more expansive genetic testing right now. So we have what's called these panels of the known genetic causes of Parkinson's disease, where there's around 26 to 30 genes that are linked to Parkinson's disease or Parkinsonism in some way. Sometimes we can do what's called whole exome sequencing. And I usually don't throw this out there because that's not where I typically start, but it's very comprehensive testing that essentially looks at every single gene in the body. And right now, it would probably be more investigative purposes for someone that has had negative Parkinson's disease genetic testing or maybe a family history of MSA. It would probably be that, we could try and see if we learn anything right now, but it might be something where we look at all of your genes today and maybe in 3 years, when we know of more genes, we can have the lab reanalyze your DNA that they already have to see if we can find an answer. So I don't know if that really answers your question. There is genetic testing that's available. It's just we might not get an answer today. It might be in the future. Any other questions? Okay, well, thank you so much. This is my contact information if you think of any questions, and I think Nicole will probably send that out too with the thank you email, but I appreciate you all having me and for your great questions.
Everyone, thank you for your time today. We will have our next session the first Thursday in January, and it will be on caregiver issues. Have a caregiver class. We actually have a caregiver class that we hold quarterly now, and we want to try it out for one of the lunch and learns. And we'll hold it on a regular basis in our lunch and learns for you, so you can have time to ask questions and everything in person or virtually. So have a great day. Thank you for your time today.